Hi everyone, it's Natalie again, and I am putting the ecologists back to back here on the No-Till Market Garden podcast. This time I talked with microbial ecologist Terry Bell. Terry runs the Microbiome Manipulation Lab at the University of Toronto, making this an international show. He came highly recommended and did not disappoint. We talk the basics of what is a soil microbiome, how can we manage the microbiome or change it, whether soil microbial inoculants work to do this, and why microbiome breeding is not analogous to plant breeding. Terry is one of those guests I had never met before this conversation, but I hope we will cross paths again in the future because his work is so cool. If you're one of those people who listens to podcasts to help you fall asleep, I really don't think this is a good choice for that because there's so much here to get the gears turning in your head. Enjoy.
Today's episode is brought to you by Real Organic Project. Does your farm deserve to be recognized for all the hard work done on behalf of the environment and your community? Real Organic Project is an add-on certification that partners with 1,000 plus certified organic farms across North America, including my own farm, Rough Draft Farmstead. Available at no cost to farmers and with minimal time commitment, it is a great way to differentiate your farm from mass-marketed corporate organic where hydroponic production and animal confinement are still commonplace. Real Organic Project is a whole farm certification program to distinguish crops grown in healthy soils and livestock raised humanely on pasture. As a farmer-led movement, we know many hands make for quick work. I hope you will lend yours by signing up for Real Organic Project certification today. Visit realorganicproject.org/no-till to apply. That's realorganicproject.org/no-till to apply. Today's episode is also brought to you by Farmer's Friend. In a world where the threat of food insecurity is quickly becoming the new norm, Farmer's Friend exists to equip, educate, and inspire growers to change the world through regenerative agriculture. Small farms are the future, and the future is now. If you need quality tools and supplies for your vegetable or flower operation, check out their growing selection of innovative and affordable tunnels, tools, tarps, and much more. Here at my home farm, we rely heavily on Farmer's Friend for our production. We have 3 of their Caterpillar tunnels. We use the silage tarps for occultation. Their row covers have gotten us through hard winters. And of course, the Quick Cut Greens Harvester has sped up our baby greens harvest tremendously. Visit farmersfriend.com/no-till today. That's farmersfriend.com/no-till. Or visit the link in the show notes and get free shipping on your entire order. All right, enjoy the show.
Yeah, so thanks for being willing to do this. I really appreciate it.
Sure, no problem.
Yeah, I saw your seminar that you gave for the AMPS department, and it was really great. So I just pulled. I tried to read some of your papers, and then I just pulled a lot of material from that talk. So, yeah. So let's get started. If you could just tell me about what you do today and how you got there, and I guess introduce yourself as well.
Sure, yeah. So my name is Terry Bell. I'm currently just starting my lab again at the University of Toronto. So I'm a faculty member at the University of Toronto, just moving from Penn State, and my lab focuses on soil microbiology in a few different types of ways, but a lot of our work is related to agricultural soils. I guess how I got here, I don't know. I started off thinking that I was going to go into writing, and then I sort of realized that I like to know what my job is every day. So I switched and moved into science, and sort of did a variety of different research experiences. I started off digging up tree roots in Finland, and then I moved around a bit and realized that I was interested in soil microbes because there's so much that we don't know about them, and because you can pretty much do whatever you want to these soil systems just to see what happens. You can't really do that if you're working with humans or large organisms. So it's kind of interesting to be able to just play around and do what you want. I've also worked in the high Arctic. So during my PhD, I worked on contaminated soils in the most northern settled site in the world. So it's a military base called Alert in northern Canada. And then, so I did remediation for several years, and then more recently have been focused on agriculture and sort of thinking about how we might be able to manipulate or shift microbiomes in soils and how that might impact function both for farmers and for just thinking about general global cycles that microbes are involved in.
What was it that kind of pulled you to agriculture?
I don't know. I mean, I think I was like getting back to this idea of just being able to like poke and prod things. I think there's some areas where people are trying to study natural systems, and they have to do a lot of painstaking work to really replicate the conditions of real life. So it's like you can't add more than this much nitrogen, or you can't, change things from what they're like in reality. What I like about contaminated soils and agricultural soils is they're kind of whatever we make them. And so there's nothing that's really totally out of the realm of possibility. So I like that there's the potential to have real impact. Agriculture covers such a large portion of the earth. It has such an important role for me, I'm really interested in sort of like conserving natural systems. And so it's sort of indirect conserving of natural systems by sort of making the agricultural footprint smaller. So I'm sort of drawn to that, but I really like that you can kind of play around with those systems a lot. And there's a lot of possibilities, basically whatever we come up with.
That's cool. I feel like I'm starting to have some insights into your personality now, which is great. I also am a poker and a prodder, so that's neat.
Good.
Painstaking is maybe not my forte.
Right.
Yeah.
Yeah, I'm not like a details person, so that's not as much for me. I like when there's a sort of big margin for error. I can just sort of like, pour some chemicals here and some other things there. That's sort of my style of science a little more.
All right. Well, so could we start off with just what is a microbiome? It's a word that maybe, I don't know, 10, 15 years ago we didn't hear much, but we hear it a lot now. So what is that?
Yeah. So I guess 10, 15 years ago is really when we started getting new tools to be able to see a lot of the microbes in the environment that we couldn't see before. So microbiology itself dates back to the 1600s, but really the way that we look at microbes now is it's this kind of science has a history of about 10 to 15 years. So microbiome is basically just all the microbes that live in whatever we call a specific sample. So if I just grab some soil out of the ground, however big that sample of soil is, I'm going to refer to the microbiome of that soil, even though in reality, that field probably continues. It could continue for many kilometers, right? So there's no real start and end, but we're just sort of calling that the microbiome of the sample we've taken. If you were to take a whole organism and sort of, I guess, mush it up and take all the microbes associated with it, you could say that you'd sampled the whole microbiome associated with that organism. So yeah, all of the different types of microbes, the bacteria, fungi, viruses, all of those things that are associated with whatever your sample happens to be.
Yeah. So when you take this new-ish technology and we say we sequence the microbiome, what are we looking at?
Yeah. So sort of classic microbiology, what we had to do was first capture things from the environment. So we would often do that by sort of creating these little vials of food. And some things would be able to eat those and grow up. If you were making, for example, you're somebody who's making a compost tea, you're sort of culturing the microbes that are able to grow in that. So the things that we're able to grow up and then poke and prod in the lab, see what they like, see what they don't like, that's classical microbiology. The sort of newer way that we're doing things in combination with that is through DNA and RNA sequencing, where we basically just sort of mush up whatever sample we collect, take all of the DNA or RNA out of that sample, and then we look at the code of the DNA or RNA in that sample to see sort of what is there, what kind of things are those microbes doing. And what that lets us do is it lets us capture not just the microbes that we could grow in the lab, but all of the other ones too. So some people estimate that you can only actually capture about 1% or less of soil microbes in the lab. So with this new approach, we're able to see far more of the things that happen to actually live in the world, and a lot of the things that are important for function in soils and other types of environments.
So how many, if you were to take a typical agricultural soil, mush it up, extract all the DNA, how many organisms might you find?
Yeah, that's a tough one. I don't think there's actually a good answer to that. One estimate I looked at recently suggested there could be up to 1 trillion microbial species. It depends also sort of what you're calling a species, what you're saying is distinct from something else. Microbes, as you might imagine, there are lots of different microbial shapes and functions and things like that, but they're more limited than say a person. Bacteria, for example, only have a single cell. There's only so many ways that they can sort of look and interact with the environment. So yeah, maybe there's 1 trillion species. Whether there's 1 trillion that we would say are really, really different from each other in terms of how they interact with the world, probably not. There's probably a number there that are very similar to each other in their function. But yeah, there's a lot. If you took one sample of soil, you're at minimum finding thousands of things in that one little sample. So a lot of different types of microbes that live together.
Yeah, and I think it's worth kind of getting into a little bit of the details about how you do differentiate. You don't typically use the word species when you're differentiating microbes, do you? So maybe talk a little bit about that.
Yeah, so there's a lot of problems with trying to define species. So one, even among sort of more familiar larger organisms, it can be tricky. But one challenge with say bacteria is that they don't necessarily need a partner to make offspring. They can just produce another cell directly. And so where do we draw the line and say, okay, this one has changed enough that we're gonna call it a new species? What we can do is we can compare sort of how similar or different the DNA in different organisms is. So if it's completely identical, we can pretty much say that that's the exact same organism. If we see some differences in the DNA, it's different than this other maybe reference organism, but is that difference translating or turning into some sort of difference in how they work? That's a harder question. So just because something is different doesn't mean that, say, from a farmer's perspective, it's actually going to do something different in the world. So we have to kind of look at it from those two perspectives of, are these things different, and do those differences actually mean something? Is it important? Are they different enough that they have different roles in the environment, or that they having one versus the other one makes some sort of visible difference to how things are working.
Yeah. And so you find all these organisms, and are they kind of just one big happy family in the soil working together to get the jobs done?
Yeah, definitely not. That's actually something that people who study this trick themselves into sometimes too. It's partly just sort of how we represent the data. So because there are so many different organisms, we can't just go and sort of, compare how abundant microbe A is across our thousands of samples. And then now we're going to look at microbe B, and now we're going to look at the thousands of different microbes that we happen to find in our samples. That's not an efficient way to understand the data. And so often what we're doing is we're representing a microbiome as a whole. So all the microbes from one sample, we can represent that as sort of a single data point and look at how similar it is to other data points. And so sometimes that sort of tricks us into feeling like, okay, yeah, this is one team, and how similar is it to another team? But in fact, every microbe that lives in the world is self-interested. It wants to grow and reproduce. Sometimes they do cooperate because it's to their benefit, but it's certainly not the case that they're all just helping each other out there. It's also important to realize, some people, when they think of microbes, they might only think of pathogens, so things that make you sick. So there are negative microbes, things that are negative for people, things that are negative for plants. there are a lot of beneficial microbes, so things that directly help people or plants, or that say convert nutrients in a way that's useful for soil functioning. And then there's a lot of things that are kind of neutral. They sort of live their lives and we live our lives, and they're not really doing anything of significance from our perspective. So yeah, lots of different microbes out there. They're definitely not a team, although sometimes some of them are able to cooperate.
Yeah. And so if you find them in this sample, you go out, you pull a soil sample, you get thousands of, we'll call them species.
Yeah.
Does that mean that all of those are active?
No, yeah, definitely not. So again, it depends a little bit on where you're pulling them from. So there's some environments where if those microbes are not active and doing things, they're probably going to get eaten or otherwise destroyed. In soil, there are a lot of microbes that are kind of just lying around. Some of them are dormant, so they're maybe waiting for better conditions. Maybe it's a microbe that it does really well when your soil is flooded and it's a low oxygen soil. And so that's a condition where this microbe can thrive. And when that condition reappears, those dormant microbes might sort of reanimate and take advantage of the situation. So in your soil at any time, the things that we see, some of them are active and doing stuff. Some of them are sort of lying around dormant. Some of them are actually dead. And the DNA from those organisms has stabilized in different parts of the soil. So we have different approaches that people are working on to try to separate those different possibilities. But it is really tricky sometimes to figure out whether the things we're looking at are microbes that are actually important in the system at the time we're looking at them, or if they're just things that are, if they're even alive, and definitely like whether or not they're things that are useful to the soil at the time we're looking at it. Yeah,
I was looking through one of your papers, and I think I read that there were some folks who were doing research on Bradyrhizobia, which are the form associations with soybeans. And and it said that they found that even in the absence of soybeans, those bacteria could survive for like over 20 years or something like that.
Right. Yeah. So I think the longevity of different microbes is still unknown. Again, part of that is just reflecting that there are so many things that we've only just discovered that they even exist. So, this summer I was at a conference. We were just talking about the rate at which microbes turn over, like how quickly how long is the lifespan of a microbe, and how long until it produces offspring, and how long until that offspring produces offspring. That seems like a really simple question, and it's something that we know the answer to for cows, for chickens, for all kinds of stuff, but we don't know that actually for microbes, how long they live in the soil, how long they persist, and when they go dormant into these sort of states that they're not even doing anything, how long can they live in that state? Maybe, thousands of years in some cases if they're undisturbed. So, that's a really seemingly basic thing that is actually really, really tricky to figure out.
Yeah. So earlier you mentioned about some traits, like there might be some microbes that are dormant, but then once there's flooding conditions and there's, not the oxygen available, they might wake up. So traits are a really important part of ecology. And for the podcast, I've spoken with a weed ecologist, and we talked about some traits that are important for kind of determining the community that arises in your field. What are some of the traits that you think about for microbes that might be in an agricultural soil, in a natural soil? Just sort of generally, what are the traits that you think about with microbes?
Sure. Yeah, I guess it depends on sort of what our specific question is, what we tend to focus on. I mean, there's some traits that are really they're really visible if you were able to actually see the microbes. So if you were to visualize them under a microscope, something like cell shape. So some are sort of more like balls, some are more like hot dogs, some are these sort of weird spirally shapes. So there's different types of ways that microbes can look, and these matter for the microbe being able to move around in their environment and things like that. We also think about traits like where microbes can live or not live. So some microbes can live across a really wide range of conditions. Some are really, really specialized, and they can only live under a really super narrow range of conditions. So we might refer to these as more generalist or more specialist microbes. We also think about things like, how quickly they can grow, how easily they can move through the world. So some can be sort of sucked up into clouds and then precipitated down really far away from where they came from. Others are going to be more bound to the soil, and they're sort of set up to stay there long term and not disperse quite as easily. So those are some of the things that we think about. And I guess from an agricultural perspective, do they perform sort of unique functions that are important for agriculture? So do they take nitrogen from the air and convert it into forms that plants can use? That's a directly useful function of microbes from a farmer's perspective. Or do they solubilize phosphorus so that plants can take advantage of that? So yeah, lots of different ways that we can think about that depending on what our question is.
Yeah. All right. So I want to talk about manipulating the soil microbiome.
Sure.
So first, how is that different from manipulating our gut microbiome? There's a lot of products out there for, probiotics and gut health and whatnot, but how is it different with the soil?
Sure. Yeah. So the idea is a little bit the same. So the types of pills you might take as probiotics, there are probiotics for agriculture. There's a large number of companies actually producing products like that. Some of them are directly applied to seed. Some of them you would apply independently. So you might apply them as a liquid to soil, things like that. There are other approaches to manipulating microbiome. So there's that sort of product-based approach. There's also this more holistic approach, I guess. So one reason that people manipulate gut microbiomes is because of a condition called Clostridium difficile. So this can happen after an antibiotic treatment. So this particular pathogen, after you've cleared out your gut, a lot of microbes have been killed off. This pathogen takes hold and sort of takes up a lot of the space and resources available in your gut. And so one of the ways that people deal with this is through what's called fecal transplantation. And they're trying to make this sort of like a more refined science, although some people take this into their own hands. They're basically just looking for anybody's diverse fecal microbiome, and you can imagine where they get that, and they insert that to try to rediversify, say, the colon. And so the idea there is that you want all kinds of either beneficial or neutral microbes to take over space from the pathogen, so the pathogen is no longer dominating the system. The closest, the most analogous thing to that in agriculture would be after people have applied, say, fungicides or antibiotics to crops, or after they have used steaming on soils, then you sort of have this void. And so possibly adding this diverse microbiome is allowing you to have a range of microbes take up space and resources so that things aren't dominated by some single microbe. One really important difference, though, between these kind of systems is that for humans, the microbes kind of come in and go out through one route, whereas in agricultural systems, there's microbes coming in from rain, there's microbes coming in from earthworms, from animals walking across, from just wind dispersal. So you have all kinds of microbes coming in all the time. And so our ability to maybe control that system is more challenging than it might be in a sort of more closed-ish system like a human gut.
Yeah, I think that's really interesting and has a lot of similarities with the soil weed seed bank, for example. Like, yes, right, there's an innate weed seed bank, but there's always going to be those wind ones that come in, and so it's impossible to close off that system and 100% manage your weed seed bank.
Yep.
Today's episode is brought to you by RIMOL Greenhouses.
Whether it's tomatoes to market, flowers in the spring, or your family's harvest, Growing in a greenhouse protected from the weather provides the right environment that you need for a harvest that you can count on. RIMOL Greenhouses are designed for durability and offer the quality that you need to grow productively year-round. Visit RIMOLGreenhouses.com to get a quote today.
If I may editorialize for a second, I own a RIMOL tunnel. I love it. We built it ourselves, and I really appreciated the quality of the tunnel as well as the customer service we received in the process, so make sure to check them out. Today's episode is also brought to you by Tilth Soil. This Cleveland, Ohio-based company is producing some of the highest quality potting mixes out there. One of the biggest lessons I learned in farming is the importance of good seed starting mixes, and that's why I included Tilth Soil in my book and have been using their seed starting mix, Sprout, on my farm since 2020. Tilth Soil produces potting mixes approved for use in organic operations like our own. Their living soil is made with fully composted food waste they collect and process themselves. Whether you need a cubic yard super sack once a year or ongoing deliveries for your year-round operation, Tilth has you covered. The team at Tilth can help with shipping, coordination, and provide ongoing support throughout the growing season. To learn more, visit www.tilthsoil.com. That's tilthsoil.com. All right, back to the show.
So if you want to manipulate the soil microbiome, say, for instance, you do have a system that's kind of got diseases or a high pathogen load, how effective is that? How effective are the markets or the products currently on the market, and why or why not are they effective?
Yeah. So the first thing I'll say is 100% there are products that have beneficial effects under the right conditions. The biggest challenge is the fact that not things can't just live everywhere, right? There's cost to an organism to be able to deal with one set of conditions and then another set of conditions. So even though there's microbes that maybe they have a wider range of soil types or habitats that they can live in, they're not going to just be able to do their best everywhere. That's just not how organisms work. And so these products are microbes that came from somewhere originally. And so probably the types of habitats they're going to do the best in are the ones that sort of that are similar to the ones that they came from in the first place. So really the biggest challenge, they've actually been marketing probiotics for agriculture since the 1800s. And the biggest problem in this entire period of time from then until now is just this unpredictable establishment issue. So you put microbes into a soil. In some cases, they're going to thrive. They're going to do what you expect them to do. In other cases, they don't do that. We had a commercial product that we were working with for a set of experiments, and we just did this sort of like pre-test to see how it was going to do across these 3 different soils that we'd collected, all from within, I think, 10 kilometers of our university. And in one case, the microbe thrived. It basically stayed at the same high level. We added to that over the course of weeks. In another case, within days, it had completely died out. We didn't see it anymore. So we get this really huge variability in terms of how a microbe is able to deal with a new environment. And that's really the challenge, because farmers want to be able to predict what's going to happen. They would probably rather have a product that is going to, contribute some smaller benefit, but all the time, as opposed to something that, maybe you'll get this huge benefit, but it's only like 5% of the time that you're gonna get that benefit. So that's really the challenge. The other challenge I would say is that there's so little regulation in some respects that you just have tons of companies, and there's not sort of enough requirement on them to sort of prove that these products are going to be what they say they are and function as expected across a range of conditions. So there's some products that we've worked with that we feel pretty good about that they can work well in the right conditions, and others that, it's not clear to us that they're going to be able to do anything. So I think there's a lot of confusion from the perspective of somebody who's trying to purchase and apply these kinds of products. So definitely I feel there's a lot of potential, but I also think that it's a really challenging thing to apply effectively right now.
Yeah, with that work you mentioned when you took the commercial product and it was able to thrive in one soil and almost completely died out in the other soil, were you able to identify or have a pretty good idea of what the differences were that led to thriving versus dying?
Yeah, we're not totally sure. I mean, one of the things that we think is a big driver of that is pH. So when we look at the microbes that live in different soils, if I was just to take a survey of all the soils across Pennsylvania, for instance, the biggest driver I would see in terms of which microbes live in one soil versus another one is soil pH. So a lot of microbes do well at sort of like a neutral pH. But then there's some that do really well under more acidic conditions and some that do really well under more basic conditions. So that's one big driver, but really it's a combination of how well adapted a microbe is to all the different conditions it's going to face in the soil. That's also what makes it complicated, because if it was just pH, you could just say, okay, well, here's our low pH microbe and here's our middle pH microbe and here's our high pH microbe and we're done. But there's also sort of like, what kind of food does this microbe need to eat, and where does it get its nutrients, and how does it deal with sort of low or high water conditions? So it's really a complex set of conditions that sort of determine whether it's going to do well or not in any particular environment.
Yeah, and so I know you have compared it to and said it's not the same as plant breeding. Can you talk a little bit about how this concept of breeding microbes or microbial communities is not the same as plant breeding, which has been very successful?
Right. Yeah. So there's a couple ways we've looked at that. So I've done some projects where we've looked at, these, you can focus on one microbe, and a lot of companies have focused on one or a few microbes, and there's a good reason for that. It's that they want to be able to sort of validate the functions of this product. So it's like, if I have this one microbe, I know that it fixes nitrogen. I can show that in a greenhouse condition, it does that really effectively. Then I have something to work from, and I can also figure out sort of why it's doing that, and maybe sort of look for improvements over time. But there's lots of functions that are additive. So there's no one microbe that does that thing. So one example would be phosphorus solubilization. Some microbes are really good at getting phosphorus from some sources in the soil. Others are able to get it from other sources. So you actually need a diverse array of microbes to efficiently extract those nutrients. And so one thing that we've done is we've looked at selecting whole microbiomes or communities of microbes over time based on sort of the highest performers. So the same way that you would select plants for breeding, you say, okay, well, this microbe is performing this function really well, or sorry, this community of microbes is performing this function really well. We're going to take that one and now transfer it forward for our next generation. But what we actually see in that case is we'll have some generations where we get this really big improvement in function, and then the next one we might get some huge decline where it's doing worse than the one that we started with. And so part of the reason I think that's so unpredictable is that there are a bunch of self-interested microbes. When you're breeding plants, all those genes are connected within the same organism. In this case, we're dealing with all kinds of things that are growing at different rates, doing different sorts of stuff. So that's been complicated. The other thing you can do is you can sort of try to breed individuals, which is something that we're exploring in my group as well. And what you want to be able to do is find traits that you can modify through time by putting pressure on them. But microbes also evolve super quickly. So if it's a trait that changes really quickly, it can also change back really quickly. And that's an issue that we've seen also. One of the things you have to do to grow up microbial products is grow them in these kind of culture conditions that we often use in the lab to study microbes. We want to grow a lot of them quickly because that's needed for sort of mass production. But what you're doing there is you're providing them all these easy resources. You're completely changing the environment. And so microbes can actually evolve quickly under those conditions. And that's something that we've seen in some of our studies, that just the growth in culture itself can actually make the microbes do worse when you then put them out in the soil. So Yeah, there's several reasons that's complicated. I do think that is a really interesting and sort of hopeful way of creating microbes that are, or sort of, I shouldn't say creating, I should say breeding microbes that are sort of more effective under the types of conditions we'd like to apply them under. But there are a number of challenges to that too.
Are there things? So this is all talking about kind of adding microbes, whether it's an individual or a community. What about just manipulating the microbial community by other management techniques, not necessarily adding microbes, but just doing something different?
Yeah. So there's a number of ways that people approach that. One is sort of through amendments like they might produce their own compost or compost teas. And so basically in that kind of approach, if you're not yourself a microbiologist, you're doing that from observation. And so you're sort of growing up these enrichments, you're adding nutrients, but you're also adding the microbes that thrive under those type of nutrient conditions. And so there may be approaches like that where you can kind of, even if you don't know exactly what microbes you're growing up, you are growing things that are doing well under the conditions that you've created. So that's sort of one way that a lot of people are doing this kind of on-farm microbial management. There's obviously ways of trying to deplete microbes. A lot of those are really blunt force. So I mentioned a couple before, things like soil steaming. In that case, you're just trying to take out everything. And that's not ideal, but if you have a pathogen situation, that might be your only option, right? Because if you have a big pathogen problem, you might end up with a total crop loss. And so you'd rather get rid of those pathogens and deal with the consequences of having eliminated a lot of other beneficial things as well. Some people are exploring like approaches for sort of more targeted subtraction of microbes. So as opposed to antibiotics that tend to target a lot of different microbes, there's another type of antimicrobial called bacteriocin. So I had a colleague at Penn State, Kevin Hockett, who works on these, and these are much more narrow and specialized antimicrobials that target very specific, potentially problematic microbes. And so people are exploring those kind of approaches too. The other thing I'll say too is that microbial management without any sort of soil management is probably not going to be really effective. There's a really interesting study from guts that basically showed that when you fed these mice what they called a McDonald's diet, so basically this low carbon diversity diet, they lost microbes over time. So they had these sort of microbial extinctions from the gut over several generations, as compared to mice that ate these sort of rich carbon diets, leading to sort of more rich carbon in the guts. So you could compare this to soil that has sort of a rich organic matter, lots of different carbon sources, versus something that's more depleted. What they found was that just spiking in new microbes didn't help with this problem, and also just changing the diet didn't help with this problem. What they needed was a combination of changing the diet, so enriching the carbon sources, and also re-inoculating the sort of diverse array of microbes. So there's a combination of things that go into having lots of microbes thrive in a soil. Part of that is sort of what the soil itself is like, because if there's nothing there to support the microbial life, you can add them in all you want, but they're not going to be able to survive very long once they get there.
This gets me to an interesting point that a lot of folks, I think, in this community do use a lot of compost inputs. And one of the things that my passion is cover crops. And so do you think that it is necessary in terms of feeding those microbes a diverse carbon diet and encouraging microbial diversity and, for lack of a better word, health? Do you think it's important to be giving them external inputs like compost, or can you achieve that just by growing a diverse array of crops and cover crops?
Right. Yeah, that's a good question. I mean, I think if you're managing cover crops in a way that they're adding organic matter to the soil, that's definitely creating a really rich and diverse environment. Plus, the cover crops themselves are going to be releasing all sorts of different compounds while they're alive that recruit a lot of things to the root zone. So one of the things that we see happen when you have cover crops in between cash crops is they're potentially enriching over the course of the year the number of sort of root associated habitats for microbes, because those cover crops are interacting with the soil in ways that are different from the cash crop. I think another thing that's complicated with diversity in general. So like we do want a diverse microbiome in that we don't know what a lot of these microbes do. And so over the course of a year, we want to say that we've got this sort of like deep pool of microbes in there, right? But when you think about how people manage all the other aspects of a farm, we're not trying to diversify plants to the maximum. We're trying to grow a cash crop really well and then use diversity in a targeted way with cover crops to add specific services and to optimize the function of the farm. And so I think there's sometimes this perception that more microbial diversity is always better. I think that the diversity in the pool of available microbes is good, and that should be sort of as high as possible. So things through like, you know, if you do farmscaping or you're sort of adjacent to more natural systems, you probably have this like deep bench of microbes that are able to contribute to soil function over time. But it may be that during the growing season, what you really need is to maximize the abundance of things that provide nutrients or that solubilize nutrients that can then be taken up by the cash crop. So yeah, I sort of went off from what you're asking there a bit, but that's, I think, another important thing to think about.
No, that's great. And that leads into another question about some work that you and your grad students have done, which was sort of looking at which are the active colonizing microbes in different places. You could explain it obviously better than I could, but that point about fungi not being quite as mobile and having that kind of diversified landscape. If you could talk about that work that you've done, and then also I'm going to throw another question on there. I think a lot of farmers are very interested in this concept of fungal bacterial ratios, and if you could talk about whether that matters, you know, and why. Mm-hmm. And also how you measure that, that would be great.
Yeah. So to the first part of your question, one thing that we talked about there was, this idea that some fungi they don't disperse as quickly or as easily as at least some bacteria. And one of the things that we've seen, so we talked before about like, you know, how effective are microbial products. One of the types of microbial products that, at least in some circumstances, has definitely been shown repeatedly to have positive effects is the inoculation of mycorrhizal fungi. So these are fungi that definitely do not disperse very easily, and they're ones that are sort of disproportionately important. So they provide essential nutrients to a lot of plants that the plants can't otherwise get. So that's sort of you know, if you're thinking about like, what are we managing here? Do we have to manage every microbe, or do we have to manage a few key microbes? That in some cases may be an important one to manage if you have a depletion of these types of fungi, if they're an important partner of the plants that you happen to be growing, that sort of thing. And sorry, the second part of your question, I've forgotten already.
Yeah, no, I know. I can only ever keep one thing in my head at once. So it's really cruel of me. So the second question was, I think a lot of people are interested in the concept of fungal bacterial ratio. Right. Some people promote that as being very important for soil biological health. So I was wondering if that is important, if you could talk about that and how you measure that.
Yeah, I mean, they have some overlapping, but some different roles in systems. So, you know, both are involved in decomposition, but they tend to, in general, focus on different components of decomposition. So definitely both are essential. One other thing is that in soils in particular and other types of dry environments, fungi can be really important for helping bacteria move around. So bacteria in the soil, ones that are sort of mobile and moving. If you've ever seen sort of an image of a bacteria with like a bacterium with like a tail-looking thing, we call that a flagellum. Those are able to move the bacteria through liquid easily. But in soil, a lot of the time, a lot of that soil space is just air. It's dried out. And so what fungi can do is they can bridge those gaps by making these networks. So some people, when they're talking about sort of till versus no till, they're talking about the fact that when you till frequently, you're disrupting sort of larger fungal networks that might develop over time. And so fungi have been shown to transport bacteria, sometimes directly, but sometimes just in association, like the bacteria on the outside of the fungi are able to find enough liquid that they can kind of move around. So the combination of both of them in soil is important. Whether the ratio specifically is important or whether that's just kind of an indicator of other soil conditions that are beneficial, I don't know. I think it's very context dependent. Fungi often do well under sort of lower pH conditions relative to bacteria. But yeah, I think they're both really essential to lots of different functions in the soil. How we measure that, one thing that we can do is we can look at sort of lipids that occur in the bodies of these organisms, and we can just look at sort of how many lipids we're coming across that are from fungi and how many that are from different types of bacteria. We can also try to estimate that from DNA ratios. So depending what kind of approach we're using, we might sort of find, so many sequences that are from bacteria and so many that are from fungi. But typically what we're doing is this kind of lipid analysis where we're looking at how many are coming from one group or the other one.
But so it sounds like, I don't want to put words in your mouth, but it sounds like there's not enough data out there to say, here is an ideal fungal bacterial ratio that you should be striving for in your soil.
Right. Yeah. And I would say that extends beyond that too. So I've had people from some sort of newer companies contact me and ask me to endorse. They basically wanted to come up with a test where they said, okay, we're going to sequence the microbiome that you have, and here's our reference microbiome. That's the best one. However they determine that, I have no idea. And we're just going to tell you how close your microbiome is to this random reference thing. But, it's very context dependent. Like if you go centimeters away from where you collected one soil, you could have really different soil conditions and really different microbes that are supposed to live there. So yeah, we definitely don't have the ability to just say, okay, at any point in space, this is exactly what your fungal to bacterial ratio should look like. These are exactly the fungi and bacteria that you should have there for optimal functioning. It's too complex to be able to say that kind of thing.
Yeah. This is why scientists are not good at making money.
That's right.
So getting back to the future of microbial products, what do you think is the future? And are you working on anything related to that?
Yeah. So I do think that they're a really essential tool, kind of. I think it's something that we haven't unlocked, but it's kind of the main part of agricultural systems that we haven't unlocked. So we know how to control the plants. We know how to control the nutrient conditions. We know how to control the water. We don't know how to control the microbes that have a lot of say over how a lot of these other things function. So they have say over whether the nutrients stay in the system or not. They have a say over sort of how well plants are able to grow and perform. So I do think that they're really important, especially, some countries are laying down more strict regulations over how much you can apply fertilizers. So like the Dutch farmer protests and in Canada as well, they're progressing to reduce sort of the amount of nitrogen that you can add to the system. And there's good reasons for that. A lot of the nitrogen that gets added as fertilizer goes to waste. It's not even taken up by the crops themselves. Whereas microbes that are able to produce some of those same nutrients. So for instance, nitrogen fixing bacteria, they might be able to produce it a little more on demand, or at least at a slower rate, so that plants are able to sort of stabilize those nutrients better, and we're going to have fewer nutrients running out of the system. So I think they're really important. I think that a lot needs to be done to figure out how we make microbes that do what we need them to do, at least for a short period, right? You don't need them to be functioning as well as possible for 52 weeks a year. We need them at least during the growing season, or maybe during critical parts of the growing season, to be doing certain types of things. And I really think that this sort of unpredictable survival issue is the main thing that's kind of limiting. So that is a really big focus of our group. We are looking at different types of approaches for breeding microbes that are more resilient to different types of soil conditions, but also more resilient to the different stages of the production pipeline. So I mentioned this idea that, like, you have to grow them up in these sort of like rich nutrient conditions so that they're ready to send out to lots of different people. And so, how do we do that and yet not have that compromise their ability to do well in a soil environment, which is much harsher, many fewer nutrients. So, big focus of our research is trying to understand how we start to focus a little more on those ecological problems, because really this industry has been mainly function focused. So it's like, we want microbes that do this, and that makes sense because that is what you want them to do in real life. So fixing nitrogen or solubilizing phosphorus, but we also need to focus on all the things that allow them to actually do well, hang on in the environment, and do the things we need them to do across a bunch of different conditions. Yeah,
you mentioned nitrogen fixation, and I know most folks are familiar with nitrogen fixing bacteria with legumes, but they're also free living nitrogen fixers. Is that, could you talk about that a little bit? And is there any focus on that?
Yeah, so there's a combination. So there's some that they have to live in association with plants. The plants themselves provide these sort of, in combination with the microbes, they create these nodule formations that allow the microbes to do their stuff under the sort of low oxygen conditions they need to fix nitrogen. There are free living ones too. There are companies that are focused on trying to figure out. So going back to the idea that microbes are selfish, they don't want to just, they're not trying to fix a whole bunch of nitrogen to help the farm. They want to fix nitrogen to help themselves. And so one thing that people are looking into is, how can we make them do more of what we want them to do, which is fix a bunch of nitrogen and not just fix the amount that they need for themselves. It's very similar to what people have done with crops, like corn, compared to where corn came from, the plants that people originally started breeding to make modern corn. It's totally different, and it's bred to meet human agricultural needs. So people are focused both on free living nitrogen fixers and those that associate with plants. The advantage of the free living ones is that they can provide benefits to any crop, not just the ones that happen to be able to form these nodules. So that is definitely a big area of investigation, both for people at universities, and companies as well.
Yeah. You talked a little bit about some of the things you're working on, but is there anything else that you're working on now that you would like to share?
Yeah. I mean, one other thing, I guess, it's sort of, I think in lots of different ways, we're trying to understand how we can take advantage of the fact that microbes evolve so quickly. So this is also why people have hope that microbes eventually will efficiently be able to break down plastics. They evolve so quickly, and now they're starting to respond to these different types of human created environments. One thing we're trying to see is, like, if we prepare microbes to use certain types of food sources, how does that translate if we then put them out in the field where there's all kinds of food sources available? Basically, like, can we tailor their preferences and then say, okay, we know you really like this. So now when we put you out in the field, we're also going to give you this. And is that going to give you an advantage over other things that happen to live there? So basically trying to see how our ability to harness their sort of rapid evolution, how we can then take advantage of that to help them do what we would like them to do when we put them out in the field.
Great. Well, it has been really interesting and fun talking with you. I appreciate it.
Thank you so much.
Yeah. And did I miss anything? And last chance, anything else?
No. I think the only thing is I didn't mention, all the people that work on our team to do this. Some of the stuff that I talked about today was the work of graduate students who have worked in my lab. So especially Laura Kaminsky and Kaylin Yates, and a postdoc who had worked with me, who's now at Cornell, Will King. They did a lot of the experiments that I sort of touched on today. But these kind of things are really big team projects, just because there are so many different aspects that we have to consider and explore. So yeah, I thought I should mention that too. Yeah,
definitely. I meant to mention it also as a former grad student. I know that it's nice to get credit for your work, and it is a team team effort.
Yeah, definitely.
Yeah. Well, I really appreciate it, and good luck in your future endeavors.
Great. Thank you so much.
As usual, thanks for making it to the end of this episode, and thanks to Terry for chatting with me. For my next episode, I'm going to stay on the soil biology theme, and we are going to talk nematodes, so stay tuned.
Hey, you all, Farmer Jesse here, just jumping in real quick to catch you up on some of what we have going on. The forum has been absolutely amazing these last few weeks, headed into the growing season. Make sure to head over there with any and all level of growing questions as they arise for you. Also, the No-Till Growers YouTube channel, that thing is back up and jamming again for another season of videos. There are new guides for onion, tomato, pepper, and spinach production. There is a seed priming primer. We were also invited by Google to beta test their new software that helps add Spanish and Portuguese subtitles and dubs to those videos. So our new videos will all have those, and slowly our older videos as well. Go over to YouTube and search for No-Till Growers and subscribe. If you'd like to support our work, you can pick up a copy of the Living Soil Handbook at no-tillgrowers.com, or one of the no-till hats, which are back in stock. We've got merch there as well. The proceeds from those sales at no-tillgrowers.com go to supporting our work, which is free and open to the public. If that work has benefited you and you have the means to pitch in, that would be super rad. Or even better, become a yearly or monthly patron at patreon.com/no-tillgrowers, where not only may you get discounts on our stuff, but at a certain level, or if you just bump up from one level to another, you get a shout out on the show. So big shout outs this week to Closter Farm and Livestock Co., Sean at All About the Garden, Bill Altman, Ian at Grindstone Farms, Stephen Smith and Oh High Roots. Huge shout out to everyone who supports our show in whatever way that you can. The Patreon page, of course, is the lifeblood of our work, so we hope you will hop on board. And that's it for me. Thanks, y'all. We'll see you next week. Bye.